Hey guys, so in this module 7, we're going to be lesson 1, we're going to be looking at cell discovery and theory. And our focus question for lesson 1 is how did the invention of the microscope lead to the discovery of cells? So, cellular discovery is well rooted in the discovery and the development of the microscope. The further the microscope has been developed, the deeper our understanding of the microscopic world has become. Now, when we're looking at the history of the cell theory, cells are so small that their existence was unknown before the invention of the microscope. Things that we take advantage, like just looking at cells or a piece of bark or something like that. Prior to 1665, nobody had ever saw this. So in 1965, an English scientist named Robert Hooke made a simple microscope and looked at a piece of cork. Now, that cork, what he was looking at was oak bark. And he was looking at these microscopic cells, and that is the term that he coined. So each one of these is actually the outline of a cell wall. Now, Hooke observed small box-shaped structures. He called these cellulae. And because they look like cells that monks lived in the monastery, that is the term that stuck. Now, from Hooke's work, we have the term cell, or the basic structure and function unit of all living things. The cell is the basis for all life on Earth. Now, the invention of the microscope has and the improvement of the instrument and new microscope techniques have led to development of the cell theory and a better understanding of cell. We have gone all the way from 1590 when a Dutch lens grinder, Hans and Zachariah Janssen, invented the first compound microscope to 2008, where we now can create three-dimensional structures and 3D views of high resolution with multiple colors and things like that. The range of which we have developed the microscope proportionally reflects the knowledge of the microscopic world. Now, the cell theory is one of the fundamental ideas of modern biology, and it includes three basic principles. First of all living things are composed of one or more cells. Everything that is alive is composed of either one cell or multiple cells combined together. Now, this is really important because Robert Hooke was the first one to come up with this theory that all living things are made of cells. Now, Mathis Schleden and Theodore Schwann, they developed a part of the theory that cells are the basic unit of structure and organization of all living things. They determined that all plants and animals are made of cells. Again, this may seem very elementary to you, but it is something that was unknown before the invention of this microscope, before people were looking at objects over and over again. Finally, Rudolf Virchow, he developed a part of the theory that states cells arise only from previously existing cells, with cells passing copies of their genetic material on to their other daughter cell. So this was one of those basic things that people didn't understand, but DNA, and this is kind of before DNA was even discovered, is what they're talking about with genetic material. Now, with this, like I said earlier, microscope technology has significantly increased our knowledge of the microscopic world. And the discovery of cells and the development of the cell theory would not have been possible without this invention. So Hooke and Leeuwenhoek would not have been able to see the individual structures within human skin cells if this microscope didn't exist. So, developments in microscope technology have given scientists the ability to study cells in greater detail than early scientists ever thought possible. Now, let's start with optical microscopes. So, an optical microscope uses visible light and lenses to magnify an object. And if we take a look at this, right, a compound light microscope is the most utilized tool in the scientific laboratory. And when we do this, we multiply the two lenses in order to get your magnification. So, for example, in this, if we take our scanning lens, which has a 4-time magnification. All right. And we are looking through that as well as a 10 times eyepiece. We multiply those two together to get our total magnification of 40. Now, as we work our way up to the next biggest lens, which has a 10 times magnification, 10 times magnification of the lens, 10 times magnification of the eyepiece, gives us a total magnification of 100. And finally, we got our biggest lens, which is 40 times objective, and multiply that with the eyepiece, which is 10, to give us a total magnification of 400, excuse me. So objects cause light to scatter, and the maximum magnification that we can actually get is about 1000. And the reason behind this is if I draw these two dots, right, we can clearly distinguish between them. But as we start to magnify even further, if I put two dots closer together and you move that object farther and farther away, eventually they then blend into one object. So magnification and the clarity of the magnification has to do with the clarity of distinguishing between these 2 points. Now, other types of microscopes we have use electrons instead of lenses, and these are non-optical microscopes. Now, in a non-optical microscope, what we do is magnets aim a beam of electrons at a thin slice of cells. And this type of electron microscope is called a TEM, or a transmission electron microscope. And this would be an image that you would see from a TEM. So because the electrons are transmitted through a specimen, they have to be dead. And they can magnify up to 500,000 times, but the specimen must be dead and stained with the heavy metal in order to see it. So it kind of limits the use of this structure. Now, a scanning electron microscope gives us a 3D image, an SEM. So an SEM, or a scanning electron microscope, is modified that directs electrons over the surface of the entire specimen, and this one produces a 3D image. This allows us to see the outside coating, the structure, what it looks like, how it bonds. And again, only non-living cells and tissues can be observed using this specimen. Finally, we have an STM, or a scanning tunneling electron microscope. Now, this involves bringing the charged tip of a probe extremely close to a specimen so that the electrons actually tunnel through the small gaps between the specimen and the tip. So in this one, we can look at things like DNA molecules, and we can see where the gaps in the DNA molecule are. The cracks and the depressions appear darker, and raised areas appear lighter. So we can actually look at very, very tiny objects. Now, if this field of study kind of interests you, you could be a cytotechnologist. And to do this, you have to have a keen eye for detail and like to solve puzzles. So cytotechnologists act as disease detectives by analyzing cell and tissue samples, often in a medical setting. The information from a cytotechnologist report can be used by a pathogen or a doctor to diagnose and treat disease. Now, a good example of this is when the COVID-19 virus first appeared. Scientists really had no idea what it looked like, what it was like. So before we could ever make a vaccine for it, we had to first identify what the structure of that object was. So they used really high-powered microscope to look at the protein structure, to look at the shape of it. How does it bond? How does it attack cells? That is the role of a cytotechnologist. So let's talk about the two basic cell types that we have. Now, all cells have at least one physical trait in common. They all have a structure called the plasma membrane. So the plasma membrane is the distinct outside layer of pretty much all of these cells. So you can kind of see this here. Here is a prokaryotic cell. You can see this outside layer that covers it. In our eukaryotic cell, you can see a very, I don't want to say similar, but the outside layer is made of the exact same type of things. Now, a plasma membrane is a boundary that helps control what enters and leaves the cell. So Cells need to maintain equilibrium. So certain things have to be able to come in freely, and other things need to be controlled on how they come in or leave. So the plasma membrane is this structure that helps to maintain that equilibrium. Now, cells generally have several common functions. And the first one is genetic material. And remember, the genetic material provides instructions for making substances that the cell needs. So remember, the genetic material that almost all organisms use is DNA. Prokaryotic cells as well as eukaryotic cells both contain this genetic material. The other thing is the ability to break down molecules to generate energy. So in these cells, energy is required. So in some way, they have to be able to form a molecule called ATP, and ATP is cellular energy. So let's kind of talk about these two basic cell types. The first one, and the one that you are composed of, is a eukaryotic cell. And these are defined as cells with a nucleus and other organelles that are bound by a membrane. So when we're talking about membranes, we're talking about a phospholipid bilayer. So eukaryotic cells have a nucleus and they have organelles, and these have these phospholipid bilayer membranes that kind of hold everything in place. The other type of cells that we have are prokaryotic cells. Now prokaryotic cells are much more simple, and they are defined as cells without a nucleus or membrane bound organelles. So this is kind of really important. Eukaryotic cells, they are cells with a nucleus as well as organelles, and prokaryotic cells do not have a nucleus and do not have membrane-bound organelles as well. Those are the defining functions or features that we have between these two cells: the lack of a nucleus as well as the lack of membrane-bound organelles. Now we're talking about an organelle. And organelles are specialized membrane-bound structures that carry out specific cell functions in different parts of the cell at the same time. So the nucleus, the mitochondria, the endoplasmic reticulum, these are all organelles that perform a specific task, almost like parts of the city. The other defining function of a eukaryotic cell is a nucleus, and it is a central organelle that controls the cell's genetic material in the form of DNA. Now, prokaryotic cells, an example of prokaryotic cells are bacteria. They're very small, very simple. But scientists think that prokaryotes are like that of the first organism. So most likely the first living thing that evolved on planet Earth were prokaryotic cells. Now, if we're looking at these prokaryotic cells, right, here is our prokaryote. Notice it is simpler and there is not much stuff going on. Up here, we have our eukaryotic cell. You can distinctly see our organelles as well as our nucleus inside of that structure. So let's draw what a prokaryotic cell is going to look like. I usually start by drawing a pill-like structure, and that's our plasma membrane. And on the inside of that plasma membrane, we have a circular DNA. So this DNA is not maintained inside of any sort of cellular structure or membrane. This is the defining prokaryotic structure. Now we also have these other structures inside here, which are called ribosomes, and these are where our proteins are made inside of the cell. And for cell movement, since they're single cellular, we also have a tail called a flagella that allows the structure to move. You can see much simpler and not much going on. Now, eukaryotic cells, again, has a plasma membrane along the outside, but their DNA is stored in a structure called the plasma membrane. And in eukaryotic cells, the DNA is linear. Okay, and again, it is stored in our structure called the nucleus. Now we have a whole bunch of other different structures, like we have folded regions that are associated with the nucleus. Some of them have ribosomes attached to them. and that is called our rough ER, and the ones without ribosomes attached are called the smooth ER. Now, the smooth ER is where our carbohydrates and lipids are made. Our rough ER is where our proteins are made. Now, we also have a structure that kind of looks like a set of smushed pancakes. All right, and that is our Golgi body. We also have where our cellular energy is produced, called our mitochondria. And again, each one of these are organelles that perform a specific task in our body. Now, the origins of cell diversity. Scientists continue to investigate why there are two types of cells. And the reason is eukaryotic cells evolved from prokaryotic cells millions of years ago. So, this is why scientists want to continue to study these different structures and how these organelles came to be. So, according to the endosymbiotic theory, a symbiotic relationship involving one prokaryotic cell living inside another, with both cells benefiting from their relationship, might have been the key to the evolution of eukaryotic cells. So, a good piece of evidence about this is mitochondria actually have different DNA than the original cell. And because of this, this is believed that these mitochondria were absorbed by another prokaryotic cell and began replicating on their own. But since they were beneficial to the cell, they were then incorporated within that structure. So, to kind of put this into perspective in the form of a timeline here, right? So, we start off with our original ancestral prokaryotic cell, and eventually, what happened, we began to have these infolding of the plasma membrane. This infolding of the plasma membrane led to the endoplasmic reticulum, the nuclear envelope, and the nucleus forming to kind of protect these different structures. Now, these simple cells then engulfed aerobic heterotrophic prokaryotes, which led to the formation of mitochondria. We also saw the engulfing of photosynthetic prokaryote. And again, this led to photosynthetic eukarya, and the ones that didn't led to ancestral heterotrophic. So again, the absorbing of these prokaryotic cells led to the evolution of our eukaryotic cell. Now imagine how organisms would be different if eukaryotic form had not evolved. Eukaryotic cells are larger and have distinct organelles, therefore can create much more complex structures. So these cells have developed specific functions, which has led to cell diversity and thus more organisms. So more complex life forms have evolved because of this endosymbiotic theory. So if we begin to take a look here, right, and we start to look, so let's compare prokaryotic and eukaryotic cells by looking at a Venn diagram. So let's start with bacteria. Now, bacteria are very simple and not very complex, so they fall under the prokaryotic cell. The next one, which is plasma membrane, remember, all cells contain a plasma membrane, whether it's prokaryotic or eukaryotic. Now let's take a look at the next view, a nucleus. So eukaryotic cells are the only ones that have a nucleus. Remember, this is the membrane structure that stores our DNA. The same thing with membrane-bound organelles, like our mitochondria or endoplasmic reticulum. Eukaryotic cells contain organelles that have a membrane. Multicellular organisms. This one is going to fall in eukaryotic cells. Okay, unicellular organisms, these ones are usually prokaryotic because, again, they're simpler, they're not as complex. And finally, do not have membrane-bound organelles. That is going to be prokaryote. No organelles. So this kind of organizes these structures, again, showing you that eukaryotic cells are much more complex and prokaryotic cells are much.
So let's kind of fill this out and relate the basic cell types by filling in the missing term. So the eukaryotic cell is larger and more complex than the prokaryotic cell. Eukaryotic cells contain a structure called the nucleus, which is a distinct central organelle that contains the cell's genetic material. Organelles are specialized structures that carry out specific cell functions. The cell, nucleus, and all the organelles are surrounded by a plasma membrane, and prokaryotic cells do not have organelles. Their cell function occurs inside the cell or in the plasma membrane. So, in summary of this unit, right, the invention of the microscope led to the study of cells. So, when the microscope was developed, we opened up a whole new world of scientific investigation, something that was once unable to be seen. The cell theorizes three principles. First of all living things are made of cells. The cell is the basic unit of structure and function, and in order to get more cells, they must come from pre-existing cells in which genetic information is exchanged. Remember, there are two broad groups of cells: prokaryotic cells, which are much simpler, and eukaryotic cells, which are much more complex. Hopefully, this helps you out with lesson one. And again, this is Mr. O'Brien signing off.